So, one of the interesting things about olfactory input is that it doesn't need to travel to an obligatory kind of synaptic, stop in a nucleus in the thalamus, unlike gustation, the other chemical sense, for example, or vision, somatosensation, audition, vestibular input, and even sort of hypothalamically detected changes in the internal environment of your body. All those have to make an obligatory synaptic stop in the thalamus before, projecting onto cortex and also receiving, lots of feedback from cortex into their specific thalamic channel, of input through the brain. So the olfactory tracts, remember, which are derived from they're the axons of the mitral cells, right? That are carrying information back from the olfactory bulbs that sit right above the nasal cavities, on the upper side of the brain side of the cribriform plate, where all those little olfactory neurons are sending their axons as part of cranial nerve number 1, the olfactory nerve. Well, these tracks are going to head back and go to a number of targets, and many of them are kind of in the middle portion of the brain, and many of them are kind of ancient structures. Some of them are cortical structures, but the cortical targets are going to be limbic targets again. They're going to be kind of, ancient kind of cortical regions, areas that are more kind of specific or idiosyncratic in how they're organized, versus the neocortex, which is the stuff we're going to see that processes a lot of visual information, for example, and some aspects of position sense and somatosensation and auditory information that surrounds the outer surface, the outer bark of the brain. That cortex is called neocortex, and we'll be talking about it a bit later. So a couple of the targets include structures like something called the olfactory tubercle, which is right where the tracks start to end, as they work their way back. It's part of what's called the basal forebrain, which is an area of the brain that's very important for cognition and memory. And then there's also a structure called the amygdala, which is a nucleus that's found sort of deep in the temporal lobe, just in front of a structure called the hippocampus. But the amygdala is really important. It's a nucleus. It's deep inside the temporal lobe, gray matter, but it responds to emotionally salient kinds of inputs, particularly those that are associated with some kind of threat or some kind of an arousing stimulus. And the amygdala can generate a powerful sort of autonomic response, can make fight or flight, can enhance. It can also orienting response, freezing response. For example, if you, and I apologize for this image, but if you open a carton of milk and you take it towards your nose, and you realize it's got that, it's rotten, it's like chunky and all that. You have an automatic sort of amygdala like arousal rejection kind of response to that kind of input. So there's very direct input into the amygdala from your nose. There are also a number of areas of midline cortex, like in the temporal lobes, that are involved with like kind of memory, particularly sort of memory for experiences, episodes, etc. Areas like the piriform cortex, that means pear-shaped cortex, which is on the medial aspect, the middle aspect of the temporal lobes. The cortex that surrounds the amygdala, so the periamygdaloid cortex, and also what's known as the entorhinal cortex. And rhinal means nose. A lot of these areas, are linked pretty directly with a structure called the hippocampus that's important for memory. And for a long time, given our understanding of the anatomy of the nose and sort of olfactory inputs into the brain, the hippocampus, the entorhinal cortex, all these regions were assumed to be just important for smell. But it turns out smell has a pretty powerful link to our emotional experiences and also to our memories. If you smell something it can take you back, if it's a scent you haven't smelled for a while. We associate these olfactory inputs pretty powerfully with specific memories, partly because of this anatomical organization of the inputs into cortex.